Let's look at the neurons, how to generate electrical signal. These neurons, they have the resting membrane potential. So that's the neurons, and because this is cell membrane, let's look at the membrane protein. This membrane protein called the sodium-potassium pump use ATP as energy source. So like every pump use electrical, use the gasoline or use battery. They're gonna use ATP as energy source. Its job is to pump sodium and potassium. It pumps 3 sodium out, so 3 positive charge ion out, 2 potassium in, so it pumps 2 potassium in. So inside the cells you have high potassium, outside the cells you have high sodium. And it pumps 3 sodium out. Sodium has positive charge, and it only pump 2 potassium in. Potassium has positive charge as well, so it's 3 out, 2 in. Every time you lose 1. And after it works for a while, you can think the sodium-potassium pump is like the AC. It runs 24 hours a day, 7 days a week. And it keeps doing, okay, eventually inside become negative, outside become positive. And if they keep doing it, will it make the inside minus too much negative charge? It's like the AC keep running, your room temperature drop from 75 to 65 to 45 until you freeze, right? The answer is no, because, well, you have those two leak channel. You have the sodium leak channel, potassium leak channel. It's like you open your window a little bit, okay, some cold air gonna leak out. Eventually they reach the balance. And you have high sodium outside, some sodium gonna leak in. And you have the potassium leak channel, some potassium gonna leak out. So eventually you reach a very good balance. And the very good balance is inside is a little bit negative than the outside. When you put the electrode, like the voltage meter, you have 2 points, right? One red, one black. And you measure the voltage difference between the inside and outside. You found inside is about minus 70 millivolt. And that's the resting membrane potential. Minus 70 milli is 0.001. So it's minus 0.07 volt or minus 70 millivolt. So inside is more negative than outside. And the neuron are able to change it, become positive. So that's the key point for today's lecture, generate action potential. Okay, so the resting membrane potential is because of the sodium-potassium pump. You pump the sodium, potassium in the opposite direction, and they don't pump them equally. They pump more sodium out than potassium in, so it will make the inside negative. And I'll leave this video to you. We'll talk about resting membrane potential. Okay, now let's look at the action potential. What happens here? In the axon here lag, that's this area. That's why I say this is important. They are able to make the inside become positive by making the ions flow in, flow out. That's why in your body you have ions, and these are the charged particles, we call them ions. And the ions have a fancy name called electrolyte. Those sports drink company want to tell you, okay, our sports drink have a lot of electrolytes. It will make you healthy. That part is correct. But electrolytes is ions. So you don't need to spend a lot of money to purchase those expensive electrolyte drink, because you can create them in your kitchen. You just take salt into water. You can share with your friend during the party. You can tell them, okay, that's electrolyte water. And it's actually salty water, salt into water. And how do you make the inside positive? These are charged particles, these ions. And when they start to flow, okay, by definition of chemistry, charged particle flow is current. And when they flow, they create a voltage difference. That's how your body creates the voltage change, and that's the action potential. So what's going to happen is they will open some ion channel. They will open the sodium channel, potassium channel. And sodium, we say sodium is high outside, low inside, right? So they're going to open the sodium channel. When they open the sodium channel, voltage gated sodium channel, so it's voltage open it. When they open the sodium channel, sodium is high outside, it's going to flow in. Every particle want to follow its concentration gradient from high to low. It's like you break a bottle of perfume, or eventually those perfume particles are going to go from high to low to the whole room. Every particle want to go from high to low. So sodium going to flow in from high to low because of sodium-potassium pump. like the high sodium. And when the sodium flow in, positive charge move in, inside become positive. So we call this depolarization. So the action potential have the depolarization part is due to the sodium flow in. Then the sodium channel closed, potassium channel open. When the potassium channel open, potassium is high inside, so potassium gonna flow out from high to low. When the potassium flow out, and the voltage drop, and that's a repolarization. So repolarization is due to the potassium flow out, and then both channel close. So this whole process take about 2 to 3 milliseconds, and this is one action potential. That's how neuron generate action potential. And because it takes about 3 milliseconds, so it can generate another one. And that's why your neuron can generate hundreds of action potential per second. And that's why they take a lot of energy, high metabolizing rate. So that's the action potential. Make the sodium flow in, cause the depolarization, and the potassium flow out, cause the repolarization. And this action potential. And this action potential is all or none. Either you create one action potential or you create nothing. You don't have 50% action potential. And this action potential can go through the whole axon, reach the axon terminal. So no matter how long this axon is, some axon can be very long, like your motor neuron. The cell body stay in the sacrum, the end part of your spinal cord. And the axon can extend all the way through your leg to control your toe muscle. It can be 1 meter long. And this action potential can go through it because it's all or none. Every time it's the same size, so it won't decay. And that's the action potential YouTube video. I'll leave it to you. I highly recommend it. Take a look, so it helps you to understand how the action potential is generated. And now it starts from the axon heel lock. It will go through the whole axon. If this axon is non-myelinated, what happens is you start the action potential here, and these channels are voltage gated because they sense to the voltage, and this voltage will open the ion channel next to it. So again, sodium channel open, sodium flow in, potassium channel open, potassium flow out, and now this action potential happens to here. It's like you take one step, and this voltage change will affect the ion channels next to it. So the whole axon are covered by those voltage gated ion channel, and this action potential start to walk. So it can travel through the whole axon. And you can imagine this is like walking heel to toe. If you want to walk heel to toe for the long distance, it will be slow. And this is those unmyelinated axon. Usually they are for short distance traveling. If the axon need to travel for long distance, they are myelinated, and that's myelin created by oligodendrocyte in the central nervous system, Schwann cell in the peripheral nervous system. And the myelin, they cover 90% of those axons, so only 10% got exposed. So the first action potential happened in the first node of round via. The gap is called the node of round via. The second action potential is not here. They can jump to 90% to the second node of round via. So the second one is here. And this time it's like I ask you, okay, walking for long distance, but you can jump. So you can run, you can jump, and it's much faster. And that's why those axons, they designed to travel for long distance, they're all myelinated axons. And when you damage this, sometimes the signal won't be able to reach the destiny. That's multiple sclerosis. So the effect is the motor control, but it happens in the neuron part. Okay, so the neuron's function, it will transport the action potential propagation. You can have the continuous propagation. It happens in non-myelinated axon. You can have the saltatory, the jumping, we call the saltatory conduction. It happens in the myelinated axon. And this signal gonna go through the axon terminal. Like from here, you'll keep going to the terminal. And this terminal gonna innervate the other neuron. Sometimes muscle, most of the time is the other neuron. So like this one, you're gonna innervate this neuron's dendrite. Most of them in the dendrite, sometimes cell body. Now you reach this area called the synapse. The synapse, they look like they physically touch, but actually there's a very small gap. This nanometer, nano is 10 to minus 9 nanometer range. The small gap is still a gap. So the electrical signal, we call this presynaptic neuron, won't be able to directly go to the postsynaptic neuron. We're not terminator. Terminator, all of them are computer chips that are electrical wired. So computer only need electrical signal only. But all the animals, we need both electrical and chemical. The reason is that synapse have a small gap. So the small gap, the electrical signal need to turn into the chemical signal. And chemical signal is neurotransmitter. I found students are more comfortable at study the chemical signal in the brain, not that much for the electrical signal. But your brain actually use both. And the next time we'll talk about the chemical signal, the neurotransmitter. We have 12 different kinds of neurotransmitter. We have the excitatory neurotransmitter and inhibitory neurotransmitter. They can make this neuron more likely to generate action potential if it is excitatory, or less likely to generate action potential if it's inhibitory, like the alcohol will trigger the inhibitory neurotransmitter release called the GABA. So it turns out you drink, you feel calm. I need a drink. I'm too anxious. And it will make your neuron less likely to fire. And it's how you feel calm. So we'll talk about this part the next time. And this area. So the neuron actually use both electrical, and all of them need to be balanced. When you have too little current, it may not be able to reach to the other postsynaptic neuron. But when you have too much, there's epilepsy, there's too much current. Neurotransmitter in the synapse, this small gap, synaptic cleft, the electrical signal won't be able to go through the synaptic cleft. So they have to turn the electrical signal into the chemical signal. That's the neurotransmitter release. And the neurotransmitter signal need to be balanced. And we have 12 different kinds of neurotransmitter. Some patients, they have less serotonin than normal, and they have the depression and anxiety. So antidepressant, SSRI, its function is to increase the serotonin stay in the synapse. Not the number of serotonin, but make it stay here for longer by inhibit the membrane protein to take serotonin back. But if you have too much neurotransmitter, it could be bad as well. It's like some people, they do drugs, cocaine, they will increase the dopamine level in this area, make you feel high. But you have some side effect, like when you feel rewarded, you eat ice cream, you get an A in this class, you feel rewarded. It also needs dopamine release. And when you do those abnormal way to make this dopamine release like crazy, and your system become less sensitive to dopamine, and turn out people do cocaine have those withdrawal effect. They don't feel like anything in their life interested them because they don't have enough dopamine compared with the cocaine's effect. So we stop here, and your test 1 covered till this part, your electrical signal. And we will talk about the chemical signal, the neurotransmitter part, for the next time.